So let me tackle viruses first. With viruses, again, here, even with viruses, the size is not important as long as you know, for example, which are some of the biggest viruses, the smallest viruses, which are pox viruses. So, for example, smallpox is caused by a pox virus, true or false? Smallpox is a pox virus, right? Which pox is not a pox virus? Chicken. Chicken is a herpes virus, right? You need to be also able to answer questions about, we talked about capsids. So true or false, all viruses have a capsid. True. All viruses have an envelope. False, right? I mean, I honestly think if you paid attention in class and took good notes, you should be able to answer them by reading the notes and not necessarily memorizing a lot of stuff. True or false, true or false, plant viruses are enveloped viruses. Or most plant viruses are enveloped. False. Why? Because to pick up the envelope, they would have to go through a cell membrane. They can't go through a cell wall. So that would be naked viruses, right? If I draw a virus like this, it's going to be nicer. Something like this. This shape is called what? Icosahedral, right? This shape is helical. Together, they're called binal symmetry. Which, true or false? This is the HIV virus. False. This is a bacterial virus, true or false? True. Specifically, these are the what? The T even. So T2, T4, T6, there's a bunch of them. Or I might ask you a question again, a conceptual one. Where is the attachment site? A, B, or C? C. Where is the genome? A. Which gets into the cell? It will have to be D, right? Because the only thing that gets into the cell is D. All of this remains outside. Right? These are guaranteed. I've done it for 13 years. I'm not going to stop. 1 point guaranteed would be the steps of a viral replication, right? Absorption, penetration, biosynthesis, maturation, release. That's 1 point out of 90. Another point, while I remember it, would be the steps in the infection the disease, steps of a disease, right? Incubation, prodrome. We'll get to it shortly, but before I forget. What other questions are there, to expect? So you have to know the difference. For example, what's a segmented genome? A genome in segments, right? What is an example? The flu virus. And please know what is the flu virus and everything about it. We discussed a lot about the So there's probably about 2 questions or 3 questions on the flu virus. So quickly, what is it? It has 8 single-stranded RNA, right? It has H and N antigens. The H are hemagglutinin, N is neuraminidase. H allows it to get in, N allows it to get out. What is the drift versus shift? A drift is small changes, which you can vaccinate every year against. A shift is a major change that reassorts the virus and you can't really vaccinate against because you don't know what to expect, right? Also, be able to answer questions like this. The cytomegalovirus has a narrow specificity, right? So meaning it can only infect a few types of cells. Cytomegalo, mega means many, so that's certainly false. The rabies virus can only infect humans. False. The rabies virus infects many warm-blooded animals, right? Which one is the only one or not the only one? Which one is the one that we discussed as a host range? Polio. Because polio is predominantly a human virus. So the steps, also HIV. So know the flu virus, no HIV. We talked a lot about HIV. What are the receptors? True or false? HIV has 2 receptors. True. And HIV infects white blood cells. True. Right? What's the difference between early and late proteins? Again, chances are you still have you haven't really devoted a lot of time for micro. But we talked about late versus early proteins, right? The early proteins are enzymes because the virus needs to replicate to transcribe. The late proteins are structural, so they are made in large quantity.